So first of all, I'd like to thank the organisers for giving me the opportunity to talk today about some of the work that we've been doing in the lab the last few years. So I've tried to pick a bit of a diversity of the type of phenotyping we do. We don't do anything really large scale, so this is small scale, sort of maybe more precise phenotyping. So I've entitled my talk today, Phenotype and Photosynthesis and Stomatal Kinetics in Leaf and Non-Foliar Tissue. First of all, just to introduce you to some of the stomatal kinetic work that we've been doing in the lab in the last few years, looking at trying to see how kinetics in stomata influence both photosynthesis and water use efficiency. And then I want to move on and talk to you a little bit about some of the work that we've been doing, exploring natural variation in photosynthesis and stomatal kinetics in a wheat magic population. And then some new work looking at ear photosynthesis and then how we're using thermography to assess some of our stomatal kinetic work. Okay, so I most likely don't have to introduce this to you, but we all know that stomata control gas diffusion into the leaf. So the stomata open and close in response to a number of different environmental stimuli to facilitate CO2 uptake for photosynthesis, but as a consequence of that, water is lost from the leaf. So stomata tend to open in response to increasing or high light intensity, and this is what I'm going to focus most of my stomatal kinetics on today. There is a spectral influence to this, which I'm not going to talk about, but is really interesting in how that alters the kinetics. And of course, lower decrease in CO2 concentration and low VPD. Whereas they tend to close in the opposite, darkness, decrease in light or high CO2 concentrations. So stomata are continually adjusting to the dynamic environment. We've heard a lot about dynamic environment so far today. And just to say, although we focus very much on light, because that is one of the most dynamic components within the environment, these signals that we've got here, they're not in isolation. So, of course, although I'll show you step changes in light, that's happening along with changes in temperature and other variables. So they're not in isolation. Really what stomata have to do is they have to take multiple signals and sort of integrate them in some sort of hierarchical response. So, and the reason why this is so important for us is the fact that stomatal conductance, or GS, is really tightly correlated with photosynthesis. So we often see a close relationship between increase in stomatal conductance and photosynthetic rate, up to a certain point, and then something else can be limiting photosynthesis. So as the stomata open, of course, they allow the CO2 in, for the substrate in for photosynthesis. But also, it's really important to remember that the stomatal conductance, the more open the stomata are, the more water will be lost. And of course, there's an order of magnitude more water lost than CO2 taken up. But this is also important for evaporative cooling and maintenance of leaf temperature. And the importance of that is really shown here on this slide from Fisher in the 1998, where he's plotted stomatal conductance against grain yield here, and you can see again there's a good correlation for these. And there's twofold reason for this. First of all, the high stomatal conductances, of course the stomata are open, they're allowing CO2 in for photosynthesis, but also these measurements were done in Mexico, and it was quite high temperatures, and that high stomatal conductance was allowing the leaf to cool down and maintain optimal photosynthetic rates, because of course the higher the temperature, photosynthesis will start to decrease. But of course that will be at the expense of water loss. So what we've been interested in the lab for the last sort of 10 years or so really is looking at stomatal kinetics and how that influences photosynthesis and water use efficiency in particular. And so this is a sort of theoretical diagram of what might happen to changes in photosynthesis and stomata with different shade flecks in this case. So the importance of this is the fact that not all shade flecks or not all sun flecks will stomata respond to, and it does absolutely depend on the duration of the change in light intensity as well as the magnitude of change. So if we look here, for example, so photosynthesis in the blue dashed line and stomatal conductance in the red line. And if we look at a shade fleck that's, say, for around about 3 minutes, a short shade fleck, and for a small duration, so say 20% change in light, we might see a change in photosynthesis, where photosynthesis drops down to the light level that's achieved at that light level, but stomatal conductance might not move or change at all. However, if we have a longer duration and a greater magnitude of change, or I should say a greater magnitude of change, what we can see is these sort of typical kinetic responses. Again, photosynthesis immediately drops down to the light level that is related to that to that light level. And stomata can have a lag phase and then slowly close. And then when the lights return back here, you get stomata can overshoot and carry on closing, and then they can open up again and that's followed by an increase in assimilation rate. And so that slow increase in assimilation rate here is most likely stomatal, as sort of diffusional constraints, but also could be biochemical components to that depending on the light level. So we've been interested in this for a number of years, looking at how that affects the dynamic input into water use efficiency and photosynthesis. So over a diurnal day, you could have lots of different sun and shade flecks. This is a typical sort of light pattern in a field. And then stomata will respond to this as well as photosynthesis. And what we've got here is two different cultivars that we've termed a fast and a slow cultivar. And these are wheat measurements that were taken. And as you can see, if we normalize that data, the effect on photosynthesis, of course, the faster ones have a much quicker time to get to, or much faster at getting to the high photosynthetic rate, and the slow ones are limiting that. And then the opposite for water use efficiency here influences that. So what we've been doing is looking at some of these stomatal responses and trying to characterize them. And so this is the typical sort of experiments that we do. So when I say this is not high throughput, it's not. It's one leaf in a chamber doing leaf type measurements with ergers. And so we put the leaf in an erger, we stabilized around about 100 micromoles of light, and then once stable, we switch the light here in the white area here to 1,000 micromoles and look at the kinetic responses of both photosynthesis here in black and stomatal conductance in the white. And so what you can see here is that when the light changes, we get an immediate jump in photosynthesis, and then this slow opening as the stomata start to open and remove that diffusional constraint on photosynthesis. And then when the light goes back down, so this is a 30-minute example, the light goes back down, we can see that photosynthesis drops to that original value. But the stomata can carry on opening, again, even though that stimuli has stopped, and then they slowly close. So basically, stomatal responses are an order of magnitude slower than photosynthetic responses. And that's very species-specific and environmental specific as well. And so what you can see here in this shaded area here in red, this represents carbon that could have been gained if we'd had speedier stomata. So if they'd responded really quickly, we could have gained some more carbon in this area here. And the area in blue represents unnecessary water loss for no carbon gain. Again, if the stomata had shut straight away, this water could have been saved in those kinetic responses. So we've been examining this for a number of years now, and on a number of different species. And to sort of parameterize this, we're using a model that was developed by Sylvain Vallée-Chambron, who's in the audience somewhere here. And previous work in this area had looked at SL max, which is the maximum slope of this stomatal response. However, that SL max is driven by the magnitude of change. So that made it very tricky for us to look at different species and how that might affect them. And so Sylvain came up with this model where it fits an exponential model. We used a time constant that we're calling K, or Ki here, that represents the time for the stomata to open to sort of 63%. So if you see K, the higher the K value, the slower the stomata. And the other two parameters I'm just going to point out is, sorry, this is sort of over the top of it, but GS min, the minimum GS, and the maximum GS as we go through these, through the next few slides. So these are the typical type experiments that we do, but with gas exchange, that's quite tricky and not very high throughput either. Okay, so I just want to first of all move on and talk to you about a project called PhotoWheat. So this is a joint project that's led by Essex with NIAB and BASF. And in this project, we're trying to explore natural variation in both stomatal dynamics and in photosynthetic capacity in wheat to try and optimize productivity. And these are co-authors with this work. So we took advantage of the magic population, the wheat magic population that was produced at NIAB. So this population has 8 fountain parents that have been intercrossed multiple times to produce this diverse genetic population with high genotypic and phenotypic variation. And this population is supposed to represent about 80% of the genetic diversity in winter wheat in the UK. So during this project, we did a number of different trials. So we did some glasshouse trials and 2 field trials looking at photosynthetic capacity, sorry, this is chopped off the bottom here, it's photosynthetic capacity, stomatal kinetics, and some anatomical features as well. So I'm going to just talk to you a little bit today about some of the glasshouse work. So we grew these in batch designs in the glasshouse, and so we vernalized the plants for about 2 months, first of all, at 4 degrees with light. We moved them into the glasshouse where we have this sort of dynamic light environment, but some supplemental light, and if it decreases too much, we grew them to growth stage 41 and then started to measure some of the photosynthetic capacity measurements on the flag leaf. And so to do this, we employed infrared gas analysis. Sorry, something completely strange has happened to my slides. Infrared gas analysis, which is, again, quite a slow procedure, but really accurate for measuring our photosynthetic capacities. And so we actually measured about 360 lines using, I think, about 10 ergers to get through these plants. And so it is possible to have some sort of high throughput with gas exchange. But I think what's really nice here is that we can see this is the light saturated rate of photosynthesis, ASAT, and Amax, the maximum CO2 and light saturated rate of photosynthesis. And within this population, we can see that there's considerable variation in both of these traits here, with values ranging from about 35 to 20 in light saturated rates of photosynthesis, which of course potentially could have a stomatal component to that, which I was really interested in. And then Amax again has a twofold variation. And so we've got normal distributions, and we're exploring these now for differences, trying to look for QTL analysis, and that's ongoing at NIAB. We also saw similar variations in the VCmax, or the maximum carboxylation rate of rubisco that you get from the initial slope of an ACI curve, and Jmax, which is the electron transport rate. And again, we've got significant variation in here that hopefully we can exploit and look for QTLs. However, what I was really interested in was trying to look at stomatal kinetics in this population. But to try and do those step changes that I talked about, looking at those kinetics, was going to be virtually impossible to do this with gas exchange. So we exploited a dual imaging system that we put together several years ago, where we've combined chlorophyll fluorescence and thermography together on a system, so we can take these measurements virtually, within seconds of each other. So this is a standard fluorimeter, and what we've done is we swapped the cameras over. We put the fluorescence camera on the side, and there's a silver mirror with a solenoid activator there that swaps between thermal and the fluorescence. So we can take a thermal image, put the mirror in place, and take a fluorescence image within seconds of each other. And when we built this dual imaging system, the idea was that we could use these two parameters. So chlorophyll fluorescence gives us an indication of carbon assimilation or electron transport, which we can sort of relate to carbon assimilation or photosynthesis, whilst thermography gives us an indication of stomatal conductance. So we have the two parameters that we need to measure intrinsic water use efficiency. Assimilation rate divided by stomatal conductance gives us intrinsic water use efficiency. And that's some of the images that we produced through this system, and these, I think, are the first images of water use efficiency that's been produced. So quite a handy screening tool for water use efficiency. Whilst we built this system, we wanted to make sure that we could control the gases within that system, so that we could, in fact, we were able to drop the the oxygen concentration to 2% to measure chlorophyll fluorescence, which means that you can eliminate photorespiration. And we did this in like a mini open-top chamber. So we had a chamber here that was built of perspex, and it had two layers to it. And you can't really see the second layer in here, but the second layer had thousands and thousands of tiny holes on it, and it sort of blew gas in. And so we were measuring both thermography and chlorophyll fluorescence in this like open-top chamber. So we exploited this and added like a clamping system onto it, so we could clamp leaves across that system, and that allowed us to measure 3 or 4 leaves at any one time under controlled gas exchange conditions. So we could control the humidity, control the CO2, etc., through mass flow controllers at the side. And so this is an example of some of our measurements. So we've got thermography here at 100 micromoles of light, we've got a dry reference in here, and then we've increased the light to 1,000. We've got the kinetic responses of that thermography. We tested the dual imaging measurements for Ki, so we estimated Ki from these thermal kinetics, and then we tested that relative to what we would also calculate using an infrared gas analyzer. And so you can see here we've got a couple of the fountain parents and then some of the progeny, and we've got a variation in Ki from both the erga and the dual imager, and we've got quite a close relationship between the two of them. So there was no significant differences between Ki, or our speed of response, in the images or with the actual LICO measurements that we used. If anything, we actually underestimate Ki in the dual imager. So what was really exciting, again, we were able to put about 360 lines through this instrument, and again, we can see that we've got some huge differences in our kinetic responses. We have some that are really, really fast here in less than 5 minutes to respond and get to maximum stomatal conductance, which is really exciting, and then some of the slow responders. So significant variation in this kinetic response. Again, we're now trying to pass these data on to NIAB to try and see if we can find any QTL analysis in them. And just to say, we also saw significant variation in GS max with these ones. We did also look at stomatal density, and I don't have time to talk to you about this today, but we saw, again, considerable variation in both adaxial and abaxial stomatal density, and we're exploring this in more detail using split chamber analysis. So if we just look at some of the relationships between some of those measured parameters, and I'm focusing mostly on the stomatal ones, is that we actually saw no association between the stomatal traits, those anatomical traits that we looked at, and GS or A, which really suggests that it's not an anatomical trait that's influencing GS or the speed of that stomatal response, which suggests that it might be biochemical, which is quite exciting because we know that there's many anatomical traits that will alter stomatal conductance, but the biochemical ones are harder to find. We also saw that the speed of response was negatively associated with the minimum value. So really it seems that you have to have the stomatal conductance, the higher the stomatal conductance at the low GS, the faster that response later on. And I think that's due to less of a lag in the phases. And of course, as we expect, we also looked at some capacity cooling measurements, which I haven't talked about, but basically, of course, leaf cooling was associated with quicker stomatal opening and greater magnitude, which I think is not really surprising. But what was really interesting was that maximum stomatal conductance, that GS max at the end of it, the saturated rate, significantly correlated both with photosynthetic capacity and photosynthetic rates, really emphasizing the control that stomata have on those rates as well as the capacity measurements. Because we used the dual imager, it meant that we also get that chlorophyll fluorescence data that goes alongside that. And we've only just started to explore this data, but again, we've got these sort of low-high light transitions, and this is an example of FQ prime over FM prime, or the operating efficiency of PS2. So we've got these dynamic components, and we've sort of characterized that into various different points on this curve, and then started to parameterize them, and we've come up with 11 different traits that we're looking at. And of course, we can explore that not only in the operating efficiency, but also in non-photochemical quenching. So we've got a huge data set here in those kinetic responses that go alongside the stomatal one. And we've started to explore, this is an example of some of the information that we've got from the non-photochemical quenching, again from our various different time points on this. We haven't got very much in induction, which is a bit sort of sad that we haven't got some sort of normalized distribution there, or the relaxation time, but we do in some of the other parameters. And there's quite high heritability in some of these traits. So we start to explore these as we have done with the other traits, and some potential QTLs are starting to pull out. But this is really preliminary data and needs a lot more analysis to go forward. So that's our real sort of high throughput or semi-high throughput gas exchange to look at photosynthetic traits. But as part of this project, we also wanted to, some take-home messages first. So from these data, I think we can see there's significant variation in photosynthetic capacity. The speeds of stomatal response and the magnitude of change are not linked with stomatal density, which I think is really interesting, and that suggests there's a biochemical component to that, which would be really nice. Again, cooling capacity is related to the speed and the minimum value, which you'd imagine. And I think what these data are really showing us is that the importance of both stomatal conductance in both temperature and gas exchange, and removing diffusional constraints on that, as well as temperature constraints. And that this natural variation in A and GS provides a novel route for incorporating these traits into breeding programs, and maybe providing idiotype-specific environments. You know, it's all fair enough saying, well, you want to have fast stomata and a low stomatal conductance to save water, but you know, not if that's going to be at the expense of CO2 assimilation rate. If you've got a situation where you can irrigate, you could then maybe have something that would lose more water and have a maximum GS with a higher temperature, or a lower temperature for that. So one key part of this work was to actually look at also ear photosynthesis. So as part of the program, the idea was to see if we could look at the photosynthetic capacity of ears. So we know that ears are green for a long period of time in the field, and it's also thought that the ear photosynthesis can contribute quite a lot to grain filling, and even more so if the flag leaf gets damaged and the ear photosynthesis becomes even more important. So we started to look at ear photosynthesis, and we developed a sort of simple gas exchange chamber that we could control the gases, put that into our imaging system, and start to screen chlorophyll fluorescence to look for differences in ear photosynthesis. And just some examples of the types of measurements we can take. We can look at the effect of awns, so you can look at the awn variety. So within that magic population, one of the fountain parents, Soissons, is an awn variety, and the rest of them are awnless. So we looked at both awns and awnless varieties. We can measure the operating efficiency of PS2 or 5PS2, FQ prime over FM prime, whatever you want to call that. But what's really nice in the imaging system as well is that we can break that down into the two sort of quenching parameters. So this parameter here, FQ prime over FV prime, is mathematically equivalent to QP. That might be more familiar with some of you. So this gives us an indication of the sort of downstream sinks for photosynthesis. So the E's with electrons are taken away from photosystem 2. And then FV prime over FM prime gives us an indication of non-photochemical quenching. So this is the maximum efficiency if all the centers were open. So any decrease in that value suggests an increase in non-photochemical quenching. And of course, if you multiply this by that, you get that. So this allows us to determine what's happening with that operating efficiency. So we could do that for these ears, and this is an example of the ear going through a light response curve. So we start with an FV over FM, and then this is the operating efficiency decreasing as the light intensity increases here. So we get a typical light response curve with the ear photosynthesis exactly the same as we do with leaf photosynthesis. And because we've done this also at 2% oxygen, we sort of eliminate photorespiration, and with some assumptions, you can sort of relate that to electron transport or relative electron transport rates, as shown in this red here. But as I say, what's really nice, and if you see here, this is these two quenching parameters that I've just mentioned. So this one in green here is that photochemical efficiency, and you can see that the shape of that really sort of mimics the changes in that operating efficiency. So it appears here that all of those changes through the light curve are mostly driven by changes in the sort of downstream sink component of photosynthesis and not due to non-photochemical quenching, which stays fairly stable. So there's an initial drop in non-photochemical quenching down here, but that stays pretty stable over the light response curve, which is slightly different to what you see in leaves. But so it looks very much like it's sink sort of downstream from photosystem 2 related to the changes in operating efficiency. Because I'm interested in stomata, I thought we'll have a look and see if there's any stomata on these organs and other parts of the leaf. And this is really tricky to see, but basically we looked at stems and relative to leaves and relative to some of these floral organs. And basically, lots of these structures actually do have stomata on them. So we've got the lemna, awns, another one of awns, and the glooms all have stomata on the leaves in fairly decent numbers as well. So I thought, well, that was really interesting. What we then wanted to know was whether these were functional. We couldn't really do that functional analysis underneath the imager, so we started to employ back-to-gas exchange, not very high throughput, but a bit more accurate for us to be able to measure some of the kinetics of stomatal conductance in these chambers. So we have some pod chambers that we've developed and just adjusted a little bit so we can fit in the ears, and we've also got a new ear chamber that's been made that has a light environment around it. This also gives us flexibility to look at different light spectra and how that might affect those stomatal kinetics, which again is something that we're really interested in the lab. So the first thing we did was exploit some of the improvements in photosynthesis that actually Steve mentioned this morning about SBPs. So Christine Rains and Stephen Drever many years ago produced some overexpressing SBPs plants in wheat, and that showed an increase in foliar photosynthesis. So we thought, well, does that translate into an increase in photosynthesis in the ears? So we put these in our gas exchange system, and you can see here this is the control plant, and then one of the lines, this SOX44, has a significantly higher photosynthetic rate in the ears as well as the leaves. So that was really interesting, the fact that you could increase photosynthesis in these as well as you can in the foliar tissue. So to look at the stomata, there's many different stomatal mutants out there, many different transgenics, and so there's one that's been a lot in the literature, which is altering the expression of some of the EPF genes that are related to stomatal patterning. So EPF1, overexpression of EPF1 tends to reduce stomatal density in leaves. And so these are plants that were made by Julie Gray in Sheffield. And you can see these are the 3 transgenic lines that have decreases in stomatal density relative to control on both adaxial and abaxial surface. So we thought, right, so we'll have a look at these and see if there's changes in stomatal density in those non-foliar tissue. And so we looked at various different components of these. The numbers are a little bit different. And what you can see is there's generally a decrease in the stomatal density in them transgenic plants, as you'd see in the leaves, but it's not always the same. And there's variation in how those APF genes are affecting stomatal density in these non-foliar tissue. So we then thought, well, we'll have a look and see what this does to photosynthetic capacity, which we expected nothing, but the stomatal density shouldn't alter the photosynthetic capacity. So we put the ears in these chambers and did an ACI curve on the plants. And again, you get this typical ACI response that you'd expect. This is the wild type up here, and then the transgenic lines are down here. And when we break these down into the two components of the ACI curve, so the slope here, which is the VC max, and this is the J max, we can see that there's very little difference between them. Apart from this one line, this OE5 line seems to have some sort of difference in the photosynthetic capacity, which you wouldn't expect to happen if you're only altering the stomata, because this is the capacity of the plant. It's not limited by the stomatal behaviour. So that's a little bit odd. But what you can also see is the differences in stomatal conductance in these transgenic plants. So we have the wild type, again, sitting up here, and then these lines all have a much, much lower stomatal conductance, driven by that change in SD, in stomatal density. So the next thing was, are these functional? And yes, they are. So we did our typical step change in the ears and looked at the gas exchange within those plants. And what you can see here, again, we've got the wild type in blue. This is our typical 100 to 1,000 micromole of light step change. You can see we get an increase in assimilation rate and an increase in stomatal conductance. And the stomatal conductances are not too bad, actually, at 0.15. They're decent conductances for non-foliar tissue. Again, we can see that the transgenic lines have a much lower stomatal conductance. And this line is really interesting. This number 2 line, it tends to have quite a low stomatal conductance, but its photosynthetic rate's maintained, which means that when we look at intrinsic water use efficiency, we have a much higher intrinsic water use efficiency compared to the wild type. But these are functional. We can see that here. We've got these kinetic responses and these changes. So literally these data were collected last week, so our next step is to try and fit the kinetic models to these and see if we can pull out some of those speeds of responses in these plants. And of course that means that if you've got a difference in stomatal density, you most likely have a difference in ear temperature. And so we've started to look at some of the temperature effects with these changes in density in steady state. So this is actually a plant at 27 degrees, and we can see here that this is a plant with a lower stomatal density. We've averaged that over several different components, and it's about 1 degree higher than this other plant here that's got about 35 millimeters per squared stomatal density. And if we think about it, we said about the stomata being important for gas exchange, but also really important for maintaining temperature. And we know that floral organs is really temperature sensitive in wheat. And so maybe manipulating stomatal behavior or stomatal densities in these organs could really be important for trying to maintain those temperatures that's required for fertilization and meiosis, etc., that are really temperature sensitive in wheat. Of course, it means that we can then use thermography as well to explore some of these kinetics. And we've got an example here again from some of the wheat. This is some of the early work where we tried to look at the kinetic responses in both the leaves and the flag leaf of 2 of our magic wheat populations here. And this was work that was done by Michaeli Farrelly. And you can see that we do see some of these kinetics, and there seems to be some sort of variation within those. But those gas exchange and dynamics for looking at the ear photosynthesis, it's again a little bit like our stomatal story. We can't go through huge numbers if we're trying to look at some of these kinetic responses. So we went back to thermography and have a system here where, this is an example of some strawberry plants under here, where we have a couple of cameras. We're in a controlled environment room and have a thermal camera here and some wet and dry references. We monitor all the conditions, and then we can do these step changes and monitor changes in thermography. So this is an example of what those images look like. So these are our strawberry plants you've just seen on the previous image. This was work done by Menji Fang, who's a PhD student with me, and he's picked out certain spots on leaves, and these data are replicated experiments as well as replicated leaves. And what you can see is we've got changes in temperature, kinetics here. And these plants have various different ranges of stomatal density, naturally driven differences in stomatal density across the leaves. And again, we can see the ones that's got the lowest stomatal density have a much lower stomatal conductance here with the kinetics and a slower kinetic response. So these are done not calculating stomatal conductance, but actually calculating what we call an IG, or a stomatal index. So it's a very, very simple calculation from thermography to get this index. Basically, using a wet and dry reference in the image, you can calculate this index value, which gives you a very rapid screen for those. So this is a really promising, rapid technique for looking at some of these kinetics. And the nice thing is that we can also do it in ears. So we've done the same thing in the ears. These are barley. We also have some barley mutants that have differences in stomatal density. And if you can just see, we can draw around some of these ears. we give that step change in light, use the temperature along with different references and taking into account that change in light to calculate a stomatal conductance index. You can see that the kinetics are less pronounced in the ears, but they are there as they were in the leaves. And so just to say that you can also, of course, use stomatal conductance to have real-time measurements of thermography using energy balance. And this was work that was done by Sylvain Vallée-Chambron, who's now here in Wageningen, when he was in my lab. And we looked at, again, this is just done on leaves across like a homemade system here, like a Blue Peter system to not to mess about with the boundary layer. But Sylvain, even though this was in a controlled environment room, and this is something to consider when you're looking at thermography, the controlled environment rooms usually have some sort of oscillating cycle as they cool and warm or sort of sort themselves out. And these are the sort of typical patterns of changes in the environment. And that, of course, will automatically alter your stomatal conductance signal from thermography. So what Sylvain did was use an energy balance along with various different reference materials, took into account these variable changes in the environment, and then plotted the actual thermal kinetics, used the thermal kinetics to calculate stomatal conductance. And if my video plays, here, this image is not a thermal image, but now this is an image of stomatal conductance, and you can see the areas that we've taken from here. This is the kinetic responses of stomatal conductance. So that means not only are you getting that temporal changes in stomatal conductance, you're also getting the spatial changes in stomatal conductance, which of course is really interesting for things like patchy stomatal behaviour and how that might be affected from different stresses such as drought, etc. So you've got both spatial and temporal heterogeneity in stomatal conductance. So maybe it's the fact that you could use the initial thermal screen as a quick screening tool to screen for changes in possible stomatal conductance using your IG, and then move to a more sophisticated system to try and nail down the exact conductances and how that might spatially be different. And the spatial information is really interesting if you're trying to alter stomatal density, because it tends not to happen uniformly across the leaf. And just to say that we've focused very much today on wheat and a bit on barley, but actually there's a lot of green material out there that's not leaves. And so we've been exploring pea pod photosynthesis, as well as some of the fruit photosynthesis here. And it seems like that contribution varies depending on the non-foliar tissue, but also how well the leaves are doing. When the leaves seem to be damaged or stressed, these organs might be really important in terms of the grain filling. So I think there's lots to exploit here in some of those tissue. So just some take-home messages. Sorry, again, this slide seems to have gone a bit strange here. I think there's significant photosynthetic capacity in non-foliar tissue. There seems to be variation in that, the same as we've seen in some of the leaf tissue. Chlorophyll fluorescence can provide a rapid screen for that gas, a rapid screen for looking at some of those non-foliar photosynthesis. Gas exchange is really useful, but something I didn't mention is that in those non-foliar leaves, like the ears, there's also respiratory CO2 that's fixed. So we're probably underestimating some of that with our gas differentials. And so really what we'd need to do is develop a new system to try and take into account how much of that respired CO2 is fixed and how much is atmospheric CO2, how important the stomata are in that as well. So we've got some ideas of developing a chamber to try and do that. We thought we might be able to do it with our 2% oxygen and our electron transport, but it's a little bit more tricky than we thought. I think that thermal imaging is a powerful approach for looking at stomatal kinetics. And I think we've shown that. It's a much more rapid tool than the gas exchange. It allows you to get through many more numbers. And I think what's really interesting is the potential to manipulate stomatal density in tissue-specific material. And I haven't had time to talk today, but we've also been looking at adaxial and abaxial surface and the importance of that for both gas exchange and cooling. And it could be really neat if we could manipulate different tissues and different organs with different stomatal densities for crop improvement. So maybe if you had more stomatal density on organs, you'd have greater cooling, which might make the photosynthetic capacity better, but also really important for fertilization. So I just want to say thanks to everybody that's been involved. So Sylvain Vallée-Chambron, who I've mentioned, is now here in Wageningen. Michele Farrelly has got a position in Italy, and Phil Davey in the lab, who does a lot of the gas exchange, and Shelley Wall was doing the anatomical mentions. So, and thank you very much for your time. Thanks.
Thank you very much, Tracy, for these wonderful insights that phenotyping can bring into these photosynthetic response. We have time for questions, actually. And as in the morning, I will ask you to stand up so that we can find you easily. I think there was a hand there first. I'm trying to figure out the order, but...
Thank you for this interesting talk, Tracy. Could you comment a little bit more about the effects of light spectra on ear photosynthesis? Have you tried anything regarding red-blue light ratios and contribution of white into these photosynthetic rates? Thank you.
So we tend to mostly focus on stomata rather than photosynthesis, but of course it does influence the amount of light that we'll be able to penetrate. On a stomatal level, we've done a lot of work looking at the blue light response in stomata, and actually have some projects where we're trying to eliminate the blue light response in wheat, because actually it's an order of magnitude greater than the red light response, but for very little carbon gain. So, for example, if you do a red light response, you have a typical stomatal conductance and typical assimilation rate. You put a bit of blue light on, you can double stomatal conductance, but barely change assimilation rate. However, it might be important in the speed of responses, so we're exploring that. So we focus much more on the stomatal side than the photosynthetic side.
So we had a question, Moshilion.
Thank you, Tracy. Very interesting talk. Regarding the ear transpiration, you show relatively high stomatal conductance at the ear level, which means you have to have a nice support of water supply there. So you should have some xylem connected to this.
Yeah.
Otherwise, you will have limitation after a few minutes. So did you check the hydraulic conductance of the ear, if
No,
it can
but
support this?
No, but I think that would be really important, a really interesting thing to do, because I said that for leaf temperature, but also the translocation aspect of trying to move things into the ear could be important as well for the importance of stomatal behaviour. So we haven't done any of that, but I think it would be really interesting to do it. So yeah,
Thank you.
I think it's a good idea.
Okay, now we had plenty of questions from this area.
Yeah, very nice talk. I have one question regarding the ETR that you calculated from the ears. Did you account for the stoichiometry of PS2 and the change in absorbance?
Yeah, usually that's my question to people. No, we didn't. So I did say relative with some assumptions. So no, that's something we will try and explore, but we've also got issues with absorption, and there's lots of issues with doing that. So yeah, you're absolutely right.
Thanks.
Then I will continue. Thanks a lot. I noticed the unit for your non-foliar tissue for the gas exchange was per square meter, and I'm wondering, is that correct? And how did you figure that out?
Yes, so we tried to, yeah, that's another tricky. We started with a 3D scanner to get the area of the leaves. So it's the area of the ears. Of course, you do then have an issue with the illumination being slightly different. So yeah, unfortunately our 3D scanner's broken. So at the minute we're trying to work it out per area on each, assuming like 4 sides. Not perfect, but yeah. That's tricky because you've also got this slight shading on each of them. So.
Okay, I think there was a third question here. If not, the mic has left already this area. Okay. One here and one here. Okay. Oh, yes.
Hi. I had a question on your validation where you checked with your gas exchange unit if it's the same. I saw you did a linear regression, but did you also look at the bias? Because it's a paired sample, I would assume?
Yes, we did. I'm saying we, McKaylee did. Do that and checked, yeah.
What method did you use? Did you use the Blunt-Altman or like a Model 2 regression or?
Sorry, I can't remember any of it, but I can get back to you. I can find that out and get back to you.
Okay, thank you.
Yeah. It's the royal we.
Okay, now we have a question, François.
Thanks, Tracy. In the MAGIC population, did the change in stomatal kinetics translate to changes in biomass accumulation or things like that?
So we've got some of those data from the field samples now, but we're just waiting for that to come through. So there's sort of ongoing analysis. I think that the first set suggested that it didn't on them. But we've got 2 years of field data, so that was just from the first field data. So,
Can you elaborate why?
Well, we had two very different field seasons as well. So we had one that was very dull and wet and one that was very dry. So I think trying to put those together, when we put all those together with the environmental data. We've also got thermography and et cetera from the field and a lot more measurements from the field data. So it's just still putting them all through the pipelines.
Yes, thanks a lot. At what time point do you usually perform your measurements, and do you look also at seasonal development of the ears or over the time period of the development of the ear?
So we haven't. That's a good idea. Again, we're sort of not high throughput, so we're sort of picking them. What we've tended to do is do the flag leaf first, then wait for the ear to come out. As soon as the ears come out, we're then trying to measure them. So, but we haven't looked at any time courses through that. That'd be something interesting to look at.
No more questions? Well, you have done pretty good after lunch. I think it was a reasonable number of questions. Oh, yes, one question from Roberto.
I can't resist, sorry, because also you brought the QTL into the picture. It's etymological question. I was a good friend with Abraham Bloom, and he liked to talk about effective use of water rather than water use efficiency. It's probably not much to do with what you showed here, and thank you very much, it was great. Out of curiosity, how are you putting that in the picture? Because I like to say farmers harvest effective use of water rather than water use efficiency.
Yeah, I think that's a really good point. Water use efficiency is a handy measurement to make in terms of but how it's valued is. And I think that's absolutely what we try and do with some of our measurements is look at the lifetime water use, using this as a trait to indicate plants that may be better at it. So for example, for the wheat, we're trying to, the rationale behind trying to manipulate those blue light responses that I was just talking about is to actually have a better lifetime water use and not waste the water. So it's there maybe at critical points such as grain film, etc. So that's very much what we're trying to do and using this as a trait to indicate possible differences that we could exploit.
Thank you.
Thank you. So Tracy, don't leave this stage too fast.
Oh.
On behalf of the organizer, there is a special gift
thank
to
you.
you. So I leave it up to you to chat with Rick about the meaning and the significance of this.
Oh, thank you very much.
But I'm sure it means something.
Great.
So
thank you.
So we can thank you and applaud again.